Hello students, welcome to my YouTube channel, and in today's lecture we shall discuss about meiosis. Meiosis is a type of cell division which takes place only in reproductive cells. Okay, yesterday in the previous video we have discussed that there are two types of cell division. First one is mitosis and another one is meiosis. Mitosis is called as equational cell division, whereas this meiosis is called as reductional cell division. Before going into the meiosis, I want to explain what actually happens in meiosis as an introduction. Say for example, there is a gametophyte, say an bryophyte thallus. So the main plant body in bryophyte is gametophyte. This gametophyte produces male reproductive structure and female reproductive structure. Antheridia and archegonia. This is the male reproductive structure and this is female reproductive structure. Male reproductive structure produces male gametes. Female reproductive structure produces female gametes. This male and female gamete fuses. Then the sporophyte grows. Sporophyte grows. Say, for example, the gametophyte, the chromosome number of the gametophyte is 4. Chromosome number of gametophyte is 4. Then, the number of chromosomes in male gametes will also be 4, and number of chromosomes in female gametes will also be 4. When the male and female gamete fuses, it gives rise to a sporophyte. Now, within a sporophyte, the sporophyte is diploid. Diploid means it is having two sets of chromosomes. Why? Why? Because it has formed by the fusion of male and female gametes. Hence, it becomes diploid, and the chromosome number of the spores which are producing here will be 8. Say for example, actually the process is these spores will undergo meiosis and the spores are released. The cells which undergo meiosis, there the chromosome number will be reduced to half. Okay, say for example, forget about meiosis. Forget the process meiosis. Don't think that the meiosis is not there. Then what happens? The spores are produced, their chromosome number is 8. Then what happens? The spores fall and gives rise to new gametophyte. Then this gametophyte is having the chromosome number 8. Then it produces male gamete, it produces female gamete. It is also 8 chromosome number, it is also 8 chromosome number. It fuses and it produces again a sporophyte. Sporophyte is having 16 chromosomes. Then 16 chromosomes, spores will come. And again gametophyte again produces male reproductive structure, female reproductive structure with the male gamete and female gamete with 16 chromosomes and fusion, and again new sporophyte, and again chromosome number is increasing. Here what is happening is the chromosome number is increasing from one generation to another generation. To cut this process, to stop this anomaly, the organisms, living organisms have adjusted or occupied, or they have developed a special type of cell division called as meiosis. What happens is, whenever the male gamete, which is having the chromosome number 4, female gamete chromosome number 4, they both fuse and they produce sporophyte which is having the chromosome number 8. When these spores which are having the chromosome number 8, before they release, they undergo meiosis cell division. They undergo meiosis cell division so that the chromosome number is reduced to half. Chromosome number is reduced to half. So the spore which is forming, it is producing the new gametophyte and it is also having 4 chromosomes, 4, 4, 4. So the meiosis helps to maintain constant chromosome number. It helps to maintain constant chromosome number. If meiosis does not takes place, then the number of chromosome from one generation to another generation, it keeps on increasing and organism becomes unstable. Such organism with more chromosome number cannot survive in this environment or in this nature. So to cut this, the organisms have developed the special cell division process called as meiosis, which takes place only in gametic cells, The cells in which meiosis takes place, those cells are called meiocytes. Now we shall discuss in detail about meiosis. Meiosis is divided into two substages, meiosis 1 and meiosis 2. Meiosis 1 is again subdivided into karyokinesis 1, cytokinesis 1. Karyokinesis 1 is again subdivided into prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is again subdivided into 5 substages: leptotene, zygotene, pachytene, diplotene and diakinesis. Then after completion of telophase 1, the cell enters into next stage called as cytokinesis 1. After cytokinesis 1, the cell enters into meiosis 2. Meiosis 2 is again subdivided into karyokinesis 1 and karyokinesis 2 and cytokinesis 2. Karyokinesis 2 is again subdivided into prophase 2, metaphase 2, anaphase 2 and telophase 2. It is again subdivided into prophase 2, metaphase 2, anaphase 2 and telophase 2. This after telophase, the cell enters into next stage called as cytokinesis 2, and after cytokinesis the meiosis is completed. Meiosis comes to an end after cytokinesis 2. At the beginning of the meiosis 1, the cell first completes interphase. Completes interphase G1, S and G2 phase. Then the cell enters into meiosis. What happens during interphase? To know this, please log on to the video on interphase. You can find the link in description. So during meiosis 1, as I told, it is subdivided into karyokinesis 1 and cytokinesis 1. This karyokinesis is division of nucleus, cytokinesis is division of cytoplasm. This karyokinesis 1, the first stage is prophase. It is an important stage. It is a very important stage in meiosis. Why? Because major events, very important events of meiosis, of entire meiosis takes place in prophase. Hence, prophase 1 is considered as very crucial stage and it takes bit more time for completion of prophase 1. So, this prophase 1 is again subdivided into 5 substages. The first one is leptotene. During prophase 1, is again subdivided into, as I told, leptotene, and the first stage is leptotene. During leptotene, what happens is nuclear membrane will be there, nucleolus will be there, centrioles which have duplicated, they starts their journey to move towards opposite poles. Their movement begins. Then condensation of DNA starts. And DNA appears in the form of sting which is having number of beads on it. Such structure is called as chromomeres. DNA is present in the form of chromomeres. And condensation is still taking place. This sting of beads is called as chromomeres. Then the next stage is called as zygote. During zygote, condensation is still taking place. Centrioles, the journey of centrioles has not come to an end. They are still they are moving. Then pairing of homologous chromosome takes place. Pairing of homologous chromosome. Homologous chromosomes are the similar chromosomes which are present in father and also mother, or which are present in male and also female. Those, say for example, chromosome number 1 of male, chromosome number 1 of a female, they both come together. This process is called as pairing, and pairing is called as a synapsis. Synapsis takes place during zygote. Synapsis is nothing but pairing of homologous chromosomes takes place during zygote. Then next stage is packaging. Still condensation is not complete, still condensation is taking place, and paired homologous chromosomes which have got paired during zygote, these paired homologous chromosomes undergo genetic crossing over and recombination. They undergo genetic crossing over and recombination. See, paired homologous chromosomes, they are undergone genetic crossing over. What do I mean by genetic crossing over? The sister chromatids, if this is a chromosome, these two are called sister chromatids. And if in a paired homologous chromosome, these two are called non-sister chromatids. These two are called sister chromatids, and in paired homologous chromosome, these two chromatids are called non-sister chromatids. Non-sister chromatids, the exchange of genetic material between the non-sister chromatids takes place. And this process, the point at which they have got attached, the paired homologous chromosomes where the non-sister chromatids have attached, that point is called chiasma. That point is called a chiasma. And after this, the cell enters into the next stage called as diplotin. Here the condensation is still taking place. And centrioles are still moving to their opposite poles. But the chromosomes which have undergone genetic crossing over and formed a chiasma, this terminalization of chiasma takes place. Chiasma starts to move towards the periphery. Just chiasma which was present, say for example, this is chiasma which has formed during Packetin stage, and during diplotin, this chiasma starts to move towards periphery. This process is called as terminalization of chiasma. Terminalization of chiasma starts during diplotin stage. And after that the cell enters into the next stage called as diakinesis. During diakinesis, nuclear membrane nucleolus is disappear, centrioles are present at the opposite poles, and the paired homologous chromosome which have undergone genetic crossing over, and here terminalization. Here during diakinesis, terminalization is complete. Terminalization is complete, but still homologous chromosomes are present together. Paired homologous chromosomes are present together. They are present at one place. They are present together. But the chiasma has been terminalized. Chiasma has completely removed. This is about the prophase 1. After completion of diakinesis, the cell enters into next stage called as metaphase 1. During metaphase 1, now the nuclear membrane and nucleolus is completely removed. Centrioles have present at the opposite poles. They have produced spindle fibers and the paired homologous chromosomes. Paired homologous chromosomes which have undergone genetic crossing over and recombination. All these paired homologous chromosomes. All the paired homologous chromosomes, they are present at the equatorial plane. They are present exactly at the center of the cell, and here this is helped by the spindle fibers which are produced by the centrioles. These spindle fibers attach to kinetochore of the centromere, and as a result all the paired homologous chromosome, they occupy the central position of the cell. They occupy the equatorial plane of the cell. This is the distinguishing feature of metaphase 1. Then next cell enters into next stage called as anaphase 1. During anaphase 1, the centrioles which are present at the opposite poles, which have produced the spindle fibers, they start to withdraw or take back the spindle fibers. As a result, paired homologous chromosomes, they get separated. Paired homologous chromosomes, they get separated. Paired homologous chromosome gets separated. See here, paired homologous chromosomes. Here centromere has not broken, but the paired homologous chromosome, they get separated and moved to opposite poles. This is the distinguishing feature of anaphase 1. During anaphase 1, paired homologous chromosomes which are formed during the metaphase 1, these paired homologous chromosomes, they get separated due by the spindle fibers. Then cell enters into next stage called as telophase 1. Cell enters into next stage called as telophase 1. As I told, during telophase 1, a reversal of prophase events takes place. Reversal of prophase events. Centrioles come to their original position. Nuclear membrane and nucleolus starts to reappear. Decondensation of these chromosomes takes place. Decondensation of chromosomes takes place. Then cell enters into cytokinesis and produces two new cells. Two new haploid daughter cells. Two new haploid daughter cells. Why? Because see here the cell is having 4 chromosomes. See here 1, 2, 3, 4 chromosomes. You see here also it is having 4 chromosomes. But at the end of the telophase 1, the number of chromosomes, they are exactly moved to their opposite position. They get reduced and they move to their opposite poles. And after the cytokinesis takes place, here the chromosome number is 4. Here the chromosome number is 4. Here also the chromosome number is 4. But here the cytokinesis, the chromosome number is 2. Here the chromosome number is 2. Now, the newly formed 2 daughter cells are having half of the chromosome when they compare to its parent cells. Now, it is having half of the chromosomes. Hence, they are called haploid daughter cells. After cytokinesis, these 2 cells enter into without any interface, these cells enter into directly into meiosis 2. Within the meiosis 2, karyokinesis 2, within karyokinesis 2, prophase 2. Here this meiosis 2 is similar to mitotic division. It is similar to mitotic division, almost similar. As the mitosis, it is having prophase, metaphase, anaphase and telophase. Here also same prophase, metaphase and anaphase, telophase. There is no any further differentiation in prophase 2. The cell enters into the prophase, centrioles which have duplicated, 2 centrioles, they move to the opposite poles, nuclear membrane, nucleolus starts to disappear, nuclear membrane and nucleolus starts to disappear. So 2 daughter cells this, and condensation of DNA takes place. Condensation of DNA takes place. Then these 2 daughter cells enter into next stage called as metaphase 2. During metaphase 2, the 2 daughter cells which are formed, the centrioles have moved to their opposite poles. Nuclear membrane, nucleolus is completely absent. Then the condensation is complete. The 2 chromosomes, here it is having 2 chromosomes, here also it is having 2 chromosomes. These 2 chromosomes are placed at the equatorial plane of the cell, held by the spindle fibers which are attached to kinetochore of the centromere. After this, these 2 cells enter into the next stage called as anaphase 2. During anaphase 2, the 2 daughter cells of the metaphase 2, there again same, the spindle fibers which are produced, the centrioles start to take back the spindle fibers. As a result, the spindle fibers, they are moving backward. I mean, the centrioles are taking back their spindle fibers. As a result, tension is created in the centromere. Tension is created in the centromere, and the centromere breaks and sister chromatids gets separated. Chromatids gets separated. During anaphase 1, paired homologous chromosome got separated. Their centromere did not break. Centromere did not break. Only paired homologous chromosomes which were placed together, those paired homologous chromosomes got separated. But now in anaphase 2, the chromosome which is formed, this chromosome, the centromere of the chromosome is breaking, and the 2 chromatids, they are getting separated. The chromatids are getting separated, and this is the main distinguishing feature between anaphase 2 and anaphase 1. In anaphase 1, paired homologous chromosome got separated. Here, chromatids got separated, and these chromatids which have got separated enter into the next stage called as telophase 2. During telophase 2, again reversal of prophase events takes place. Reversal of prophase events. Nucleolus, nuclear membrane start to reappear. Centrioles come to their original position. Centrioles come to the original position. Decondensation of chromosome starts. Decondensation of chromosome starts, and it forms DNA, and again it enters into cytokinesis 2, and finally 4 haploid cells are formed. 4 haploid cells are formed during the meiosis. The very important point here is meiosis started with the single diploid. Diploid means 2 sets of chromosome. One pair is coming from male and another pair is coming from female. Having 2 sets of chromosome, it started with the diploid parent cell. At the end, it has produced 4 haploid daughter cells. 4 haploid daughter cells. Okay, meiosis gives rise to 4 haploid daughter cells, whereas mitosis it gives rise to 2 diploid daughter cells. This meiosis is called as reductional cell division. Why it is called as reductional cell division? Because chromosome number is reducing. It is getting divided. It is reduced to half. Chromosome number is reduced to half. Hence, this meiotic cell division is called as reductional cell division. Hence, this takes place only in the gametic cells to maintain the constant chromosome number. To maintain the constant chromosome number. This is about meiosis. Thank you.